Light scattering occurs when a particle passes through a laser beam. Forward scatter signal is detected just off the axis of the laser beam in the forward direction. Side scatter signal is collected at approximately 90 degrees to the laser beam. Forward scatter light is based on two properties: size and refractive index. Click each property to view its effect on forward scatter. The larger the cell size, the more forward scatter light is generated as a cell or particle passes through a laser. Refractive index is a physical property that indicates how light is transmitted and causes light to bend and change velocity as it passes through different materials. The larger the difference in refractive index between the cell and the surrounding sheath fluid, the more forward scatter is generated. Side scatter light is based on the granularity or internal complexity of a particle. Click granularity to view its effect on side scatter. The more granular the cell, the more side scatter light is generated as a cell or particle passes through a laser. A cell's size, refractive index, and internal complexity contribute to the light that is scattered as a cell passes through a laser beam. Proteins on the cell membrane, the number of nuclei, and any granular materials or organelles inside the cell affect light scatter. Click each cell type to view its effect on light scatter.
Forward and side scatter.
Forward and side scatter can be used to distinguish different cell types in human blood. In this plot, 3 blood cell populations can be identified based on their light scatter. Each dot represents a cell, and its location in the graph corresponds to the amount of light scattered by the cell. With the use of antibodies, distinct cell populations can be identified by the markers present on the cell surface. For example, T cells exhibit CD3 proteins. Monoclonal antibodies for CD3 may be used to distinguish T cells from the other blood cells. When a fluorescent dye is conjugated to a monoclonal antibody, it can be used to identify a particular cell type based on the antigenic markers of the cell. Adding conjugated antibodies to a mixture of cells or particles is referred to as staining or labeling. A flow cytometer can detect blood cells based on the presence of monoclonal antibodies with bound fluorochromes. A fluorochrome is a functional molecule that fluoresces or emits light. How does a fluorochrome fluoresce? When a fluorochrome is excited by an energy source, such as a laser beam, the fluorochrome absorbs the energy and then emits energy of a higher wavelength. For example, when a FITC fluorochrome is excited by a 488 nanometer laser, emission of wavelengths at approximately 520 nanometers occur. Each fluorochrome can be characterized by its unique excitation and emission spectra. The excitation spectrum indicates the range of wavelengths that will excite the fluorochrome. A 488 nanometer blue laser is within the FITC excitation spectrum, so excitation of FITC occurs when it passes through the blue laser. The emitted wavelength is always lower in energy than the incoming excitation energy. Common fluorochromes include FITC, PE, per CP Psi 5.5, PE Psi 7, and APC. Click each fluorochrome to view its excitation.
Fluorochromes are often bound to multicolor antibodies to enable cellular identification.
Fluorochromes are often bound to monoclonal antibodies to enable cellular identification. The more binding sites or markers on a cell for a particular antibody, the brighter the fluorescence intensity, and the higher on the fluorescence intensity scale the data appears. Cells that have the CD3 antigen will bind CD3 FITC antibodies and are said to be CD3 positive. This example shows cells that have been stained with CD3 FITC antibodies. Cells with CD3 FITC antibodies will appear farther to the right on the FITC scale. Cells and particles have a small amount of autofluorescence. More than one fluorochrome can be used simultaneously to identify cells in a sample tube. In this example, each fluorochrome shown is excited by a 488 nanometer laser. Notice that the emission spectra of the fluorochromes overlap each other and bleed into the detection filters for other fluorochromes. This phenomenon is known as spectral overlap. The bars on the graph indicate the range of wavelengths that pass through filters in front of the FITC, PE, and per-CP detectors on several BD cytometers. FITC emission spills over into the detection filters for PE and per-CP. How does spillover affect data? Due to the spillover of FITC fluorescence into other detectors, the FITC population appears to be positive for PE and per-CP. Notice that the displacement of the FITC population in each graph corresponds to the amount of spectral overlap between FITC and the other fluorochromes, PE and per-CP. Compensation allows you to distinguish cells or particles that are truly positive for specific fluorochromes from those that appear to be positive due to spillover into other detectors. In this example, the FITC population appears to be positive for PE. Click the buttons to view the effects of applying compensation to correct for spillover. Notice changes in the location of the FITC population and the median PE value of the FITC population in comparison with the negative population. Compensation enables the use of multiple fluorochromes with overlapping emission spectra within an experiment. Different cell populations can then be identified using different fluorochromes. This example shows a dot plot with 3 cell populations. Cells with CD3 appear on the right of the APC axis compared to cells without any CD3 binding. Cells with both CD3 and CD4 appear in the upper right quadrant. Reflect on what you have learned in this module. What factors affect light scatter? How are scattered light and fluorescence used to detect populations? Why is it important to know excitation and emission spectra for the fluorochromes in use?